My name's Ben Forbes. I'm a Professor of Pharmaceutics at King's College London, and it's a great pleasure to welcome you here today for this morning session, this workshop, which is on behalf of the Association of Inhalation Toxicologists. It's a workshop concept that was put together by Joe Kilgore, who's speaking today, Simon Moore, who's also part of the organising committee of the Association of Inhalation Toxicologists, and then myself as well. And the purpose of today's workshop is really to give everyone an overview of the process by which we assure the development, the safety during the development of inhaled medicines. So we've put together a short programme. We've got some specialists in various aspects to talk you through what's done, why it's done, the way that it's done. Clearly, it's a huge challenge because it's a massive area. It's a challenge for our speakers to try and give all the content they could give in their short talks. So we'll try to keep to time. We'll try to give you key messages. And then what we've configured is a panel discussion at the end of the morning. So if there's anything that's not being raised or anything that begs a question whereby you'd like a little bit more detail, you can either ask it in that session or obviously you could follow up with any of the speakers after the conference. So all the presentations will be available post-conference on the DDL website. So no need to take notes. You can sit back, relax and listen. And without any more delay listening to me, I'd like to pass over to my co-chair, who will introduce the first speaker.
Thank you, Ben. I'm Victoria MacDonald. I'm an independent inhalation toxicology consultant. I'd like to introduce our first speaker, Carsten Erhardt. Carsten is a professor in pharmaceutics and biopharmaceutics at the School of Pharmacy and Pharmaceutical Sciences in Trinity College, Dublin. And Carsten studies the disposition and the epithelial transport of drugs following their administration to the lungs using in vitro and ex vivo models with interest in molecular origins of airways disease. Carsten has numerous scientific publications and he serves as editor of various pharmaceutical journals, and he hosts a workshop on pulmonary drug delivery which alternates between Dublin and Istanbul.
Thank you very much. I hope you can see me. I can't see anyone. This is a terrible stage. So introduction is short. I have no idea about toxicology, but that's apparently okay according to Ben. So I was asked to give a general introduction really about the field, about targets. So I thought I'd start very gently talking about inhalation drug delivery in general, talk about the lung, physiology, how we get airway aerosols delivered, and then give you some examples for targets and therapies, both for local as well as systemic delivery. So here's just an overview. Do we have a pointer here somewhere? This just says back and forth.
Not that I know. I'll try and find you one.
Okay, anyway, so just gives you some ideas, some of the highlights, perceived highlights of inhalation therapy going all the way back to before the Dark Ages. So inhaling drugs is not a novel concept. It has been around for a while, but it really, probably in the last 50, 60 years, it gained much more importance with the introduction of proper inhalers like PMDIs. Now, we all have a lung, and it's the most important organ of the body, and I can come back to that and give you more details on why I believe it's the most important body later on. And the physiological role of the lung is gas exchange. So it's about 10,000 or 11,000 liters of air going in. At the same time, from the vascular side, there's about 3,500 liters of blood being pumped through the lung every day. Anatomically, the lung can be divided into two sections, the conducting airways with the trachea all the way down to the bronchioli or small airways. And then also we have the alveoli, which is the gas exchange or peripheral lung. In the proximal lung, we have an epithelial layer which has cilia on top and a mucus blanket. And this is where particles that are inhaled and are deposited there are trapped and then are moved up and swallowed eventually. And also, you can probably see here, there are large blood vessels of the bronchial circulation, as well as cartilage rings to stabilize the whole system. When you go further down in the small airways, you see the cartilage has disappeared and has been replaced by a smooth muscle layer. And this is where some of the receptors, for example, for bronchodilators, are localized. So if you want to target a beta-2 receptor with a bronchodilator, you have to reach these small airways. Bless you. The airway epithelial layer has become a bit flatter, but still it's a monolayer of epithelial cells with cilia. There's still mucus on top of that. And again, these are the areas, the conducting airways, that are most often targeted when we talk about local delivery. If you go further, you reach these kind of sponge-like structure, the alveoli, and at a further magnification, you can then see here there's an alveolar type 1 cell. That's a capillary cell. In between there's a basement membrane. And from the air side to the blood side, we're talking of, there's a distance of probably 300-400 nanometers. So it's an extremely thin barrier that allows for perfusion of gases, O2 in, CO2 out. At the same time, it's tight enough to prevent leakage of water, because there's, of course, a lot of water liquid in the capillaries, and if something leaks out, there are lots of ion channels to transport sodium chloride and water back. And if this is disturbed, you get an edema, which is a big problem. Okay, now, take home message 1. If you're not good with numbers, think about tennis court. So I guess you have seen this in one or another, guys. So the total surface area of the alveoli in a human, in an adult, is anywhere between 100 to maybe 140 square meters. That's the size of 1/2 of a single player's tennis court. And that is of interest because this massive surface area can also be utilized for systemic drug delivery. So we're going to come back to that at a later point. So depending on your delivery system, on your formulation, on your inhaler, on your inhalation maneuver, you can either target the central airways or maybe the distal airways, and you can either get a more central deposition, a more localized action, or you can probably target the distal airspace for systemic delivery. All I'm saying today is about humans. I know you're a toxicologist and you rarely work with humans. You work with rats, mice, ferrets, dogs. Humans can inhale and will inhale sometimes when you tell them to do so in a certain manner. A rat won't. So just to bear that in mind. So replicating inhalation maneuvers in animals is a bit more challenging. So that's just another electron microscopic image by Ewald Weibel that shows you the surface of a human alveolus. You can see here, this is a type 1 cell. The type 2 cells that generate surfactants are much smaller, but number-wise, there are more of those. And you can also see this dense network of capillaries that's omnipresent here. Okay, so we do not only have epithelial cells. There's also lots of immune cells present in the lung. They are dendritic cells, they are macrophages. And that is important because you might generate an immune response, and this is either a problem if you generate antibodies and then go into an allergic reaction eventually, but it could also be wanted in case of administering vaccines down the airways. So just to mention that. And as far as clearance is concerned, because the lung is important, it has a very efficacious set of filters to keep it clean, because we only have one lung, and this has to last for 70, 80, 90 years. We have, as I mentioned earlier, the mucus that is moved by cilia, so the mucociliary escalator in the upper airways, and in the lower airways we have the macrophages. So your drug, deposited as a particle or as an aerosol, has to dissolve to reach its receptors, and if it doesn't dissolve in time, it will be carried away. So designing particles for long-term release in the lung, let's say a PLGA-based microparticle that releases the drug over 2 days, is rather pointless because that particle won't be in the lung anymore after 2 days. Similarly, if you target the lower airways, the alveoli, you have to consider that your particle will end up in a macrophage, and that may cause the macrophage to come up with some reaction which might not be what you like to see. Okay, also important to bear in mind is there are healthy lungs and there are diseased lungs. In most cases, when we talk about local treatment, we are treating lungs that do not look like the ones I just showed you. This is a healthy airway. This is a healthy airway with some aspirated mucus, and these are COPD or asthma airways. So they have a very different physiology, and that also means they might have different deposition characteristics when it comes to aerosols. So just to summarize the first points, so the physiological function of the lung is gas exchange. We have a large thin surface area in the alveoli. There's lots of filters there. We have very efficacious clearance mechanisms that are different in the two sections. We have mucociliary escalator macrophages. We talk about an immune active tissue, and of notice, a diseased lung looks very often very different from a healthy lung. Okay, now, why do we want to do pulmonary drug delivery? First of all, it gives us a reason to attend this conference, right? So DDL, no pulmonary drug delivery, no DDL, big bummer. So apart from that, if you inhale something, you bypass the gastrointestinal tract. Why do you want to do that? Well, if you have a drug that has issues with degradation, because it's very sensitive, for example, to enzymes, or has really poor absorption characteristics, for example, proteins, peptides, antibodies, all those things, when you ingest them as a tablet, they usually do not work very well. Also, if you have drugs that have pharmacokinetic problems, because they are heavily metabolized or transporter substrates, or their interactions with food and so on. In that case, you might consider pulmonary delivery as an option, or of course, if you want to deliver the drug to the lung. It usually makes more sense to inhale because you get a higher concentration, you have a high local bioavailability. That means you need generally a lower dose when you inhale, and there are some examples, I didn't bring the slides, but when you, I think for pirfenidone, IPF drug, when you inhale, you get an 800 times higher local concentration than when you take the tablet. So that clearly makes sense in that case. And if you have a lower dose, you can also assume that you have fewer side effects, which from a toxicology point of view is certainly desirable. You have a very rapid absorption and action that can be wanted. If you want a sustained delivery, inhalation is not really what you want to do. You can tweak around a little bit with the formulations, you can come up with liposomes, but a real sustained delivery for more than a day or so, I would not recommend going by inhalation. And it's non-invasive. So if your alternative is an injection, most patients would appreciate inhalation. If your alternative is a tablet, I'm not aware of any patient who likes to inhale if they can take a tablet. So it really depends. It's a good thing, but it's not necessarily a great thing if there are better alternatives. There are issues. We can only inhale a certain amount, and I just put up here about 100 milligrams or so max. So if you have a drug that needs a higher dose, then inhalation is a problem. You have to always consider local effects like bronchospasm. You always require a formulation, and you always require an inhaler. And that, of course, is much more development issues. If it's an aqueous formulation, it has to be sterile, which again has issues when it comes to production. The regulator, and we probably hear about that much later in the conference, is always a bit more skeptical when it comes to inhalation, and all of this amounts to higher costs. So when it's about, especially here, systemic delivery, getting a foothold in the market and defining a niche that generates profit might be an issue, because especially when it comes to systemic, there are, of course, other routes of administration that might be not as great, but also work and are much cheaper to achieve. So I won't dwell on this. There will be other talks, but there are aerosol-related parameters when it comes to deposition delivery, and there are patient-related parameters. For example, particle size, shape, and so on, inhalation volume flow rate. Just to give you a brief example, if you inhale through your mouth for 4 seconds, and here, and I guess you've all seen these kind of diagrams at one form or another, here's the deposition, here's the particle size. You can see that in the upper airways, large particles are deposited at a high rate. In the conducting airways, it's kind of the intermediate size, and then if you want to reach the lower lung, you're probably looking at a size of about 2 to 3 micrometers. Now, if you inhale through your nose, deposition is much, much worse from a delivery point of view, or much better from a filter point of view, because the nose is a very good filter. So inhaling through the mouth makes total sense. And these data here are shown for an inhalation volume of 1.5 liters and a flow rate of 45 liters per minute. If you change those parameters, this will change, as I show you here. So this is just a model done with a software called Prelodium. So for different inhalation volumes and flows, you can see those different lines here. And this is alveolar deposition. So you can see it's always a maximum of about 2 to 3 micrometers of particle size, but there's also some variation here when it comes to inhalation flow and inhalation volume. So if you take a slow, deep breath, you can target the alveoli. If you have a fast inhalation, you probably target more centrally low bitter particles. So that always needs to be considered. So pulmonary delivery works both locally as well as for systemic, and you can tweak this. We have a respiratory tract that is designed by Mother Nature to be kept clean, and these filters and clearance mechanisms don't differentiate if it's a drug or an inhaled fly when you cycle. The nose is an additional filter set that you are probably all using unless you have a cold, but most of you are breathing through your nose right now. I'm talking, so I'm breathing through my lungs, so I have much less filter capacity, but I'm already sick, so that's fine. And if you want to target the central lung, you should be anywhere between probably 7, 8, 5 micrometers. If you go further down, you should be below 3, but not too small because nanoparticles are exhaled. And inhalation flow rate, volume, and particle size are all important. And is, of course, potentially highly variable from person to person and affected by disease state. Okay, now, local delivery. This is just something you can look at in detail when you download the slides post-conference. This is just something I took from a paper which gives you target attributes for inhaled compounds, and this is for locally acting drugs. So in many cases, you see the opposite of what you see in an oral drug. So you could generally say, as a rule of thumb, if you have a compound that is really bad as an oral drug, it might work very well in the lung. If you have a very well rule of 5 embracing compound with solubility and all of that, and no transporter interactions, then there's no point inhaling it. It will be pretty rubbish because it will be cleared from the lung rather rapidly. So you want something with low oral bioavailability. It can be lipophilic and all of these things. So what compounds are we looking at? So this is from the same paper that was published about 5 years ago. And initially, we were only looking at targeting lung disease. And this, of course, started with asthma. Eventually, COPD was added. And if you look at these first 3 generations of drugs, they're really, well, short-acting, longer-acting, and ultra-long-acting, but they all do the same. They're either bronchodilators, starting with short-acting beta agonists, and then we got the LAMAs eventually added to it. But there's really not a lot of innovation here. Molecules were tweaked, they were made better and longer acting, but it's always the same target. So for many, many years, for the first 50 years or so, almost inhalation therapy, we're always doing the same stuff, which was a bit boring, really. And now on this slide, I kind of summarized indications that already exist or are currently being developed. I'll give you some examples. So in asthma, again, we have the bronchodilators, we have inhaled corticosteroids. They've been there, but there really isn't a lot of new development in that field because I think it's pretty much maxed out. The compounds we have work, it's great. COPD, some of the same compounds, bronchodilators are used. Corticosteroids is a bit, not really sure if the benefit is there. And a couple of years ago, the first PDE4 inhibitors were introduced, but those at the moment are all given orally. So there's 1 or 2 developed for inhalation, but I'm not sure how these programs will succeed or not. And then, of course, eventually cystic fibrosis with inhaled antibiotics. Tobramycin started and was a huge success and was really a game changer in inhalation or in generally in therapy of cystic fibrosis. Mucolytics are also inhaled. We now have an approved COVID-19 vaccine for inhalation. And, well, this is, you can argue if pulmonary arterial hypertension is a lung disease or not. It affects the the blood vessels of the lung. It affects the heart. It's kind of all the same compartments. So I mentioned it here. So inhaled iloprost and treprostinil are approved, at least in the US. So these therapies are available. And in these fields, there are also lots of new developments going on, as well as I think the hot areas at the moment really are pulmonary arterial hypertension, interstitial lung disease like IPF, bronchiectasis, and also lung cancer. There are lots of things in the pipeline in preclinical, also clinical development, looking at those diseases and various targets, mostly done by smaller companies, probably a risk mitigation strategy of large pharma. And again, this is not the most up-to-date picture. I added bits and pieces. So these are some targets that are currently being investigated for asthma, COPD, and so on. And again, just I like to dwell on this. So if you look at leukotrienes, there's montelukast available as a tablet. If you go to PubMed, you see there are quite a number of papers where academics try to come up with inhaled formulations of montelukast. There's a reason why it's available as a tablet, because the receptors for the leukotriene D4 specifically are almost exclusively expressed in the lung. There are some in the spleen, but all of the others are in the lung. There is really, from my point of view, no reason to develop inhaled therapy, because you will unlikely have any systemic side effects if the receptor is nowhere else than in the lung. So in that case, go with a tablet. So publishing a paper on making a microparticle for montelukast, you can do it, but it's probably of limited value. Anyway, I touched on this earlier. So ILD, just an example from a talk I gave on a different occasion. So there are drugs already approved for inhalation therapy of interstitial lung disease, treprostinil, but that's specifically approved for pulmonary arterial hypertension associated with this, but also inhaled pirfenidone and nintedanib, galectin inhibitors, RNAi against MMP7, and anticalins. They're all in early stage clinical trials. So there's a lot of interesting new developments, and there is even more, of course, in the preclinical pipeline. So this is really an interesting space to watch. Similar for PAH, lots of new targets out there, lots of developments happening at the moment. So if we switch gear briefly and talk about systemic delivery, in the good old days when Ben and I were young, I used to have this slide where I said, okay, you can deliver locally, you can deliver systemically, and local is for small molecules and systemic is for large molecules. That slide, I think I can bin, and I did. So again, similar to before, these are indications that require a quick onset of action. A quick onset of action means, oh, pulmonary drug delivery, we get something in, we get it absorbed quickly, and then it works. The ones where we have brackets, we have products on the market, like diabetes. There's an inhaled insulin out there for, well, almost 20 years now. Forgot to close that bracket. First approved after insulin inhaled drug as a small molecule, loxapine, which is also misspelled, for agitation treatment in schizophrenia and bipolar disorder. And also there's an approved levodopa for off episodes in Parkinson's. So those are all small molecules. And well, insulin is a different story. I'll come back to that in a second. But there are lots of other indications, again, where drugs are in the pipeline in clinical trials for treatment of systemic conditions by inhalation. So in this case, we would mostly talk about an intact lung and just using it as an entry pathway to the system. So I just want to briefly touch on inhaled biologics. So no one wants to do that, right? Unhappy child, unhappy stuffy here, probably a horse. And if the alternative is injection, then inhalation might be interesting. So biologics. You can deliver drugs to the site of action in case of vaccines or nucleic acid therapies, antibodies, and that means you need a much lower dose. Or you can deliver them systemically. In the past, especially when we talked about insulin, growth hormone, calcitonin, and all of that, systemic delivery was the thing. If you go to conferences now, there is not a lot of discussion about systemic delivery. All the delivery is about local, all the things. You need a lower dose of an expensive product, so that makes total sense. And of course, you have lesser side effects if you have a local delivery. And it's a non-invasive alternative, as I already pointed out. and it usually provides a fast onset of action. If you don't want that, there are means with pegylation and so on to extend that a little bit, probably adding enzyme inhibitors, but generally fast onset of action is what you get. So the first inhaled biologic indeed was for local action, pulmozyme. And this is an example that has been out there for 30 years that you can still make a lot of money. So $500 million in revenue for Genentech is nothing you can sneeze at. And we talk about a rather large molecule, and in the development it was discovered that you get a sizable bioavailability inhaling this rather chunky molecule. This then gave rise to the idea to inhale proteins for systemic delivery. Sorry, let's stay with this new slide. Other things that you can do locally, and there's just an example here. You can inhale heparin. This was used quite successfully in a phase 1 study in Turkey during the COVID pandemic. So severe COVID patients were, half of them were treated with inhaled heparin and the others with standard of care. And you could see that there was a much better outcome for the heparin. So heparin had a positive effect on COVID. And you can also inhale vaccines for a local immune response. So this works. You get a mucosal immune response. And since COVID, we all know everything about it. And what is probably a bit under the radar in Europe is that in China, they approved this Convidicia Air last year, and they already vaccinated 200 million people with this. And it's kind of, I don't know, over here, not really that well documented, but what they do, they have this tap, they fill a plastic cup with an aerosol, and then the patient has to inhale from this plastic cup. So pretty much going to a Starbucks, getting your coffee and get your vaccine shot. So this apparently works pretty well. When we talk about systemic delivery, there was Exubera, which was a dry powder inhalation. It worked pretty well. There were lots of issues regarding the device, costs of goods, what have you not. It resulted in one of the largest write-offs for pharma industry in history. It was on the market only a very brief amount of time. And I guess you've seen all these things if you have one of those inhalers. And there's lots of others I could share you. There's a lot of add insult to injury. So it was a good product, but it didn't sell. And Afrezza became available a couple of years later and is at the moment the only inhaled insulin on the market. In the US, in Brazil, phase 3 in India, it uses again a dry powder. The inhaler, the Dreamboat, is about the size of my thumb. You have differently filled cartridges. So it's a very snazzy device. The bioavailability is much better than of Exubera. And it is also, if you consider the price that you pay in the US, actually now cheaper if you break it down to units than injection. Nonetheless, I'd like to be a bit heretic here. Yes, it generates net revenue, but $13.5 million in 3/4 of a year is not a blockbuster. So, I mean, it's more than the year before, and it's definitely more than when it started, but it's not a lot of money. So you wonder, is this really worthwhile doing, right? So summarizing, there's not a lot of innovation going on in the classic indications in asthma and to a certain extent in COPD. There are many more targets and new drug candidates in other diseases like lung cancer, ILDs, PAH, and infectious diseases. And some of them are in development also for inhalation therapy. And when it comes to biologicals, delivering them to the lung is at the moment much hotter than delivering them through the lung or via the lung. And with that, I'm done. And as the chair already pointed out, we have a fantastic workshop. You're all invited to come next year to Dublin. And
thank you very much, right? Carsten. That was fantastic.
Great, thanks.